Swayam Prabha. Digital India. Educated India.
Hello, my dear students. Channel number 11, course title Medicinal Chemistry. Lecture title Physicochemical Principles of Drug Action, Part 1. Faculty name Dhamak Vikrant Murlidhar. Module number 4 and lecture number 6. DTH Swayam Prabha, sponsored by MHRD New Delhi and conducted by IIT Madras. So let's see the contents of today's lecture. Introduction to physicochemical properties of drug. After that, we will be looking towards the concept of pharmacokinetics and bioavailability. So these concepts are related with physicochemical properties of drug. It does not directly deal with physicochemical properties, but they are interrelated. So firstly, we should know the meaning of pharmacokinetics and bioavailability. After that, we will look into the concept of solubility, dissolution, acid-base properties of drug, and partition coefficient. So these are 4 physicochemical properties we will be conducting in these lectures. So let's see firstly an introduction to an physicochemical properties. The ability of chemical compound to elect pharmacological or therapeutic effect is related to influence of various physical and chemical properties of chemical substance on biomolecules that it interact with. So here a chemical substance means a drug. So it has certain physical and chemical properties, and it has to enter into lot many barriers in the body, and after that it shows some chemical interaction with a biomolecule. So biomolecules are nothing but we will say it's an like proteins, or commonly we will call it as a receptor. So interaction of that chemical substance with receptors, so that will show an pharmacological and therapeutic effect. So it all depends on its physical and chemical properties of a drug. So first part, that is physical properties, and next that is chemical properties. So both these are interrelated with each other, because a chemical substance or a drug substance having a chemical structure has certain physical properties. Physical property of drug is responsible for its action, and chemical property, the drug react extracellularly according to simple chemical reactions like neutralization, chelation, oxidation, and later on it will interact with the biomolecules. Biomolecules nothing but receptors. So we'll see the concept of pharmacokinetics. So there are two important terms in case of drug action. One is pharmacokinetics and second one is pharmacodynamics. So pharmacokinetic means what body does with the drug, and dynamic means what drug does to the body. So we'll be watching pharmacokinetics over here because it is important to study physicochemical properties of drug. So this is a human body. After taking a drug from inside, so it will get absorbed. So we'll see one by one concept. What are these concepts? So firstly, drug will get absorbed from a site. Once it is been absorbed, so it will be distributed. So this is nothing but a distribution of drug in the different compartments or towards different tissues. With the help of transporters. After that, next step, that is metabolism. So how does a drug break down? So in some cases, drugs are active before metabolism, and in some cases, drug becomes active after metabolism, and all this process it takes place in the liver, or it has been preceded by some enzymes like cytochrome phosphate. And lastly, drug will be excreted. So drug, it has been eliminated after its action, and drug can be eliminated by certain ways. So these are all things happening in the pharmacokinetics. Shortly it is said to be ADME: absorption, distribution, metabolism, and excretion. So we'll see now the concept of each individually. So absorption, it is a process of movement of unchanged drug from site of administration to systemic circulation. So it is important to understand over here it is that it is unchanged. So drug property should not be changed, or there should be no alteration in the structure, and the drug should remain unchanged from site of administration. So site of administration, it can be oral, it can be rectal, it can be parenteral, it can be transdermal. So drug, it reaches to a systemic circulation. So it means that it reaches towards an bloodstream for distribution. So this is the first process in case of an pharmacokinetics. Second, that is distribution. So it is a reversible transfer of drug between compartments. So compartments over here, so we already in the earlier lectures also we have seen how compartments or how bilayers are. So these bilayers, they does not allow any type of a molecule to enter easily. So it requires certain properties, then only it will be coming towards and site of action for an showing an pharmacodynamic effect. So it is important to understand that it is reversible transfer. So it means that it maintains an equilibrium, and as it maintains an equilibrium between the two compartments, so drug, it is been distributed into different tissue compartments. So it is a crucial stage to reach a drug towards a site of an action. After that, next step, that is in metabolism. So chemical conversion of one form to another. So it means that there is a change in chemistry or change in chemical structure of a drug, and it is due to show some effects or to nullify the effects or to make a compound more active, or it may become inactive. So metabolism, so as per desired, so metabolism works. So either after metabolism drug becomes active, or in case of some drugs, after metabolism drug becomes inactive. And last step, that is excretion. So it is removal of drug from the body, and in between this, all this, it shows an pharmacodynamic effect. It is after distribution or it is before excretion. So in the third process, it shows an dynamic effect, pharmacodynamic. So what does drug do with the body? So it shows an pharmacological action. So these are four important steps of an pharmacokinetics. So here one should understand that when drug it is more hydrophilic, or we can say when it is more ionic, so it is easy to so it is not easy to absorb, because whenever there is a second part that is of a distribution, so drug should be having some lipophilic characters, or it should be lipophilic to pass through and compartments, because for distribution, a drug should be an lipophilic, and because the bilayer between, or phospholipid bilayer in between, so they are made of a lipophilic part, and that is why drug should be a lipophilic to enter into the compartments. If drug is highly ionic, or we can say if drug is highly hydrophilic, then a drug it will be unable to enter into an compartments, and as there will be no entry, so it will not show any action. So here it is important to understand that whenever drug it is been designed, so it should be designed in a such way that a drug should be, some partly it should be an ionic, and some partly it should be lipophilic, or we can say hydrophilic and lipophilic balance should be adjusted. Because if drug it is only an lipophilic, so there will be no metabolism, because for metabolism, or we can say when there is interaction with an receptors, so drug should have some such type of an molecules in between that it should interact with an receptor. Because earlier also we have seen that in case of an receptor binding, so it shows some type of an hydrogen bonds, or some ionic bonds, or some covalent bonds in between, and for that purpose a drug should have some ionic part also, and with that it should have a lipophilic part to get been distributed between the compartments. And for if drug it is highly lipophilic, so there will be no metabolism, because enzymes whichever it will act in the liver or anywhere in the body, so they will not convert the fully lipophilic drug into an ionic form, and it should have a balance. And metabolism is such a process that lipophilic molecules, or we can say lipophilic as well as an hydrophilic molecule, it converts to an highly ionic molecule, and once it converts to an highly ionic molecule, so it get dissolved, or it gets solubilized into an water, and as it has been solubilized into water, so drug will be removed through an excretion. If drug it is high lipophilic, so it will circulate into body through a different compartment, but it will not come into a phase of an excretion, because for phase of an excretion it requires an ionic molecule, or it requires an hydrophilic molecule, and once it becomes an hydrophilic, then only it is possible for a drug to come into an water or get solubilized into an water. So ways of excretion, so it may be through facial, so it may be through urinal. So for that purpose a drug should be metabolized, and as it has been metabolized, so it get converts to an ionic form, or we will say it get converts to an hydrophilic molecule, and as it becomes an hydrophilic, then only drug will be excreted. So in case of a drug designing, so one needs to understand all these four concepts, because there should be maintenance of an lipophilic and hydrophilic balance for absorption and distribution, and also for metabolism, and lastly it should be an hydrophilic after metabolism, or there is certain chemical conversion or change in structure or alteration in structure to make it ionic by using certain enzymes like cytochrome phosphate, and to excrete the molecule from a body. So it terminates the action of a drug when it has been excreted through the body. Now, second concept related with this physicochemical properties understanding, that is bioavailability. So name itself indicates that availability. So it is dealing with an available form, and bio because a drug should remain unchanged. So bioavailability is defined as rate and extent of absorption of unchanged drug from its dosage form and become available at site of action. So dosage form here it means that drug it is embedded in the form of tablet, capsule or any other, and it should be available at site of action, but condition is that it should remain unchanged. So drug should remain unchanged because once it changes the properties or once it changes the chemical structure, so it will be unable to mimic the action or give an action towards a receptor. So importantly, drug should remain unchanged from site of absorption and be available at site of action. So we will be understanding with the help of diagram. So here it is drug, and once drug it has been absorbed, so it get dissolved, and some part of drug it remain undissolved, or in some cases it remain undissolved, and all it happens in contents of stomach and intestine. So it has to go through a barrier of an acidic environment or basic environment in case of intestine and acidic environment in case of a stomach. So drug has to drug after absorption has to come into an GI tract, and once it comes to an GI tract, firstly it will come to an stomach, and after that intestine, acidic environment, and after that an basic environment. Still some drug remain undissolved, and once it comes to an gut wall, so drug it is ready for now distribution towards and different compartments, and non-absorbed drug, so it will be eliminated or it will be removed from the gut wall, and after that it will come into a liver, and once it comes to a liver, or we will say after distribution, so once it comes to a liver, so there will be metabolism, or we can say there is some alteration in the structure. So either it becomes active or either it becomes inactive. So it is according to a prerequisite of an desired drug what we have designed it accordingly. And once it pass through a liver, so it also come across an first pass metabolism, or we can say loss of drug during first pass of a liver. So it is that once a drug it enter into a liver, so it get metabolized due to some enzymes. So some of enzyme like liver microsomal enzymes, so it causes an first pass metabolism, and due to this first pass metabolism, drug becomes inactive. So drug which remain unchanged, so there is no change of drug from the first phase, right? And drug remain unchanged till the liver, so it is available, or we can say bioavailable dose fraction. And drug which has been undissolved during the dissolution phase and which remain unabsorbed during the absorption phase, and which has been metabolized during the first pass effect, it is said to be bioavailability losses, or we can say a drug which is of no use now, and only this bioavailable dose fraction which will be responsible for an activity in the body. So we'll come towards first physicochemical properties, that is solubility. The solubility of substance at given temperature is defined as concentration of dissolved solute which is in equilibrium with solid, solute. Here terms like temperature, because a temperature it affects and solubility. As the temperature rises, there will be difference in solubility, means it is in direct proportion that solubility increases with an temperature, and so that is why here a term temperature it is been used, and it is at defined concentration. So some of an concentration, or we can say particular solute, it has been dissolved into a solvent, so that is said to be an solubility. And there are some other factors also that on what solubility depends. The solubility of drug depends on nature of solute and solvent as well as temperature. So already we have discussed temperature. After that, pH. So some drugs are solubilized at particular pH, and after that, pressure. So pressure is also one of the things that it increases the solubility of a drug. So for solubility, or for an interaction of a drug with a receptor, so it must be in the form of a solution. So once a drug it enters from an absorption site, so it forms a solution, and before forming a solution, so it disintegrates into an smaller particles, and once it has been disintegrated, so it dissolves and it forms a solution, and once it forms a solution, so it is ready for interaction with the receptors. So all the time, so it should be in solution form or it should be in a liquid form to interact with the receptor. So no solid drug can interact with the receptor, or whenever there is a transport of a drug from across the membrane, so it should be in a solution form, so then only it can come towards a site of an action or to interact with the receptors will be available. Next concept, that is dissolution. Dissolution, a process in which substance form a solution. The dissolution of drug is important for its bioavailability and therapeutic effectiveness. So to bioavailability, earlier also we have seen that, so drug it should be in unchanged form till the site of action from where it has been absorbed or from where it has been entered into one body. So it should remain unchanged, and then only it will show an therapeutic effectiveness. So for that, it has to cross, or it has to go through a dissolution. Dissolution rate is the speed at which solid dissolves into a solvent. So drug entering into a body, if it is a solid, so it should get dissolved into a solvent to show its effectiveness. In some cases, like we are taking oral liquids or by parenteral route, so at that time a drug it is in the liquid form, so there is no need of a dissolution, so it will directly get solubilized into a body fluids and it will show an action. So diagrammatically we will now understand what is actually a dissolution. So here you can see a drug, or it is in dosage form over here, that is tablet, and it disintegrates. So disintegrate, it means it forms in small particles, and once it forms in small particles, so it is ready for dissolution. So dissolution, it is nothing but the small particles disaggregate and they enter into a solution. Solution means solute particles, they are being dissolved into a solvent. So solvent here it is nothing but, and these are fluids in the body, and drug get dissolved into the fluids of an body, and after that it it will be absorbed, and drug will be ready for entry into blood and the body. So whenever there is designing of a dosage form, not a drug. So dosage form it is, and we can say an embedded form of an drug. So which will show, or which will act through some process, and it will show an action. So importantly, it is having a drug inside it, but a dosage form it is little bit different thing. So it is having some cofactors, or we can say some co-drugs which are not showing any type of an activity. So they are said to be other ingredients like additives, and important that it is having a drug that is said to be an API, active pharmaceutical ingredient. And so due to some physical and chemical uh, properties, or to enter to an systemic circulation, so drug it should be in a proper form or in a proper dosage form, that it should not overcome first pass metabolism in the liver, or it should it should be uh, it should be easy for this ADME, that is pharmacokinetic parameters. So that is why it is been it is in the form of a dosage form. So these are two crucial stage, and these are in process quality control parameters also for drug designing in case of an not drug designing in case of an dosage form, that at what time it disintegrates and at what time it dissolves. And these are two in process quality controls, or quality control test for an dosage forms, and after that it will show an action. Now third property, that is acid base properties of drug. So many of the drug they are being classified as acids and bases. As it is noted shortly, a large number of drugs can behave as either acids or bases as they begin their journey into a patient in different dosage form and end up in systemic circulation. So drugs can be acids or drugs can be bases, but there are some drugs which shows both the properties. So according to the environment, it shows both the properties. So because acids, so when the favorable conditions, it is for an acidic conditions, so it shows an acidic property. When favorable conditions, it is for bases, so it shows an basic properties. Because a body or a GI tract from where it has been absorbed, so it has a different pH conditions accordingly, and due to different pH conditions, so it will show a different characteristics of an acids and bases. And this happens due to because the complex structure of drug molecule, it contains both the things which are responsible for an acidic condition as well as an for basic condition. So we'll see detail an example. Drugs acid base properties can greatly influence its biodistribution and partitioning characteristics. So biodistribution, it means distribution into the tissues, and partitioning, it means drug should enter through a lipid bilayer, and it depends on the acidic and basic properties of the drug. So during the drug designing, so it is important to understand the acid base properties and a site of action where we are desired, and lipophilic and hydrophilic balance is also important. But with this lipophilic and hydrophilic balance, it should one a scientist should consider for an acid base properties of drug, because it is responsible for biodistribution and partitioning characteristic, and ultimately lastly it results into an uh, therapeutic effect of any drug. So there are lot many theories, or basically there are three theories to explain acids and bases. So they have defined acids and bases as per three theories. So one of an theory, that is RNS theory, second one is Lewis theory, and third one, that is Laurie-Bronsted theory. And also there are some other theories which have explained what is acid and what is base. But in case of an physicochemical properties of drugs, Laurie-Bronsted theory, it is one of an most suitable theory for acid-base behavior, rather than having that RNS theory, or we can say RNS theory, it is related with H plus ions and OH minus ions, while Lewis theory, it is related with electron donation and electron accepting capacities of a molecule. But most suitable, that is Laurie-Bronsted theory. So it is related with an proton acception and proton donation. So according to that acid base, it is been defined. So according to this theory, Laurie-Bronsted theory, acid is any substance capable of yielding a proton, means donating a proton, and base is a substance which is capable of an accepting a proton. When an acid gives up a proton to base, so it is converted to its conjugate base. Similarly, when base accepts a proton, it is converted to its conjugate acid. So we'll see. So here it's an example of an acid and base. So acetic acid having an H plus ion and water, so which is acting as a base over here. So this one reaction, it is reversible reaction, and now it forms a conjugate base and as well as an conjugate acid. So molecule which acts as an acid donating its proton is, after a reaction, it forms an conjugate base. So a molecule which is an acid at first instance, so it becomes base later on. So that is nothing but an conjugate base, and this water which is acting as a base, now it becomes H3O plus. So it becomes an conjugate acid. So why it is said to be conjugate acid and conjugate base? Because in the reversible reaction, so it will be able to donate a proton, and this conjugate base, it will be able to accept a proton. So this is why it is said to be an conjugate base, and this one is said to be a conjugate acid. As the definition says that acid is any substance capable of donating proton. So it donates the proton, that is H plus ion, right? And it forms a negative charge, and this negative charge, it is formed because oxygen, it is more electronegative, and it takes away an electron from a bond between hydrogen and oxygen and takes electron towards an oxygen, and now it forms an H plus ion which has been donated towards an base. So base is nothing but substance capable of an accepting an proton. So this negative oxygen accept this positive hydrogen, and it forms an H3O plus. So it is indicating an conjugate acid, and reversibly, if reaction takes place, so it acts as an acid because now it is having an available proton to donate, and it is proton deficient, or we can say it is able to accept an proton from this acid. So it is said to be conjugate base as well as an conjugate acid. So this is one of only an acceptable theory for physicochemical properties of a drug, or so now we'll see properties of ciprofloxacin. Earlier also we have talked that some of the compounds which act acid as well as in base, and it is due to their chemical structures. So ciprofloxacin, it is one of an antibacterial agent, so it binds to an DNA gyrase enzyme and it shows its antibacterial activity. So ciprofloxacin, it is one of a complex molecule, and the molecule contain multiple functional groups and therefore possesses both acidic as well as in basic properties. So this acidic and basic properties, they are due to this multifunctional groups present in it and which are capable of showing an acidic as well as in basic properties. So depending on what location molecule is, so it will show acidic and basic properties. Depending on pH of solution or tissue, so if we are doing an activity outside the body, so then we can say depending on a solution, and when it is inside the body, so depending on the pH of tissue. So differently the pH of a tissue changes in the body as per the location where the tissues are. This molecule will either accept a proton or donate a proton. So these are two responsible groups, that is secondary alkylamine and showing an acidic properties, that is carboxylic acid, which is responsible for its donating properties, or we can say acidic properties, and alkylamine, it is responsible for accepting properties or basic properties. And so apart from this, there are some other groups which are responsible not for an acidic basic properties, but which are essential for entering the molecule into an lipid bilayers or coming towards an site of an bilayers. So that is why a compound is said to be an amphoteric, as like water, it acts as acids as well as in base. So depending on what molecule it interacts, so it will act acid as well as in base. So similar type ciprofloxacin is that it is amphoteric in nature because it is having a multiple functional group which are responsible for acidic as well as in basic properties of ciprofloxacin. So this is a structure of an ciprofloxacin. This is a basic nucleus of an fluoroquinol. So a fluorine was and halogen when it was been introduced. So after that, ciprofloxacin shown an activity, or the fluoroquinolones showed an activity, because this is responsible for it makes a molecule more lipophilic, and which is responsible to enter into an lipophilic membrane. Earlier molecules were not easily entering into lipophilic membrane, but as fluorine it was being introduced at this position, so it was passing a lipid bilayer. After that, there is one more, that is ketone, and ketone is also a neutral center over here. This neutral center, it is responsible for hydrogen binding properties, as it is having an oxygen which is able to donate an electron towards an hydrogen, and it is responsible for DNA interaction also, DNA gyrase interaction. So that enzyme, it binds and it forms an hydrogen bond with this also. Next, it is carboxylic acid. So the drug, it is showing an acidic properties due to this carboxylic acid moiety, and which is responsible for its acidic character. After that, there are certain groups which are responsible for its basic properties, like this arylamine chain, which is having an cyclopropene group, or which is having an lipophilic group. Sometimes here, in case of some molecules of this fluoroquinol, so it is having an C2H5 structure or lipophilic chain, and cyclopropene, it has shown more activity or prominent activity. But this arylamine, it is not available easily, because it is not having an exchangeable hydrogen atom or exchangeable proton, and that is why, so it is a weak base. It is having only this two lone pair of an electron over a nitrogen. After that, similarly, this nitrogen, it is even embedded between this quinolone ring and piperazine ring, and it acts also as a weak base because it is not having an hydrogen for an exchange. But this arylamine, which is in piperazine, so it is responsible for this basic properties of a molecule. As we have seen earlier also, that molecule either will accept a proton and secondary alkylamine. So this is nothing but a secondary alkylamine, and all these are tertiary amines. And this one is only a group which can act as a base, or which will show an basic properties compared to these two groups, because these are basic. These are basic, but these are weak one, and it is little bit strong, this arylamine chain. And this one is secondary structure because it is having an hydrogen from one side, but these structures, they are having an tertiary type, so it is not having an hydrogen. And when it acts as a base, so this nitrogen, it will have one more hydrogen over here, so that is, it accepts an proton. When it accepts an proton, now this nitrogen, it will have a positive charge, and after that, donation of an proton or donating an proton, so this group, it is responsible for donating of an proton, that is carboxylic acid group, and it will form a negative charge over here, because hydrogen, when it will leave, it will give away a lone pair of an electron towards an oxygen because of its electronegative character, and that is why it shows a negative charge over here. So this group, it will be said as a conjugate base once it loses an hydrogen, and this group, it will be said as an conjugate acid, because when reactions are reversible, so it will be able to, this arylamine will be able to donate, and this will be able to accept after the reaction. So these two are responsible mainly for acidic as well as in basic properties, and rather these two are responsible for entering a molecule into a lipid bilayer. And also this lipophilic region, it is responsible for entering molecule into a lipophilic biomembrane, and also this piperazine ring, it is responsible for lipophilicity in the molecule. So from this structure, it is also been understood that when molecule it is been designed, so it should have in certain properties, not only having an lipophilic characters, but also an ionic characters, or compounds which are making compounds soluble into an we can say water or body fluids, but there should be a maintenance of an lipophilic and hydrophilic balance when molecule it is been designed. So this molecule, it is perfect for an interaction with an now receptor, but here you can also see that there is maintenance of an lipophilic as well as an hydrophilic balance. So these are the groups like cyclopropane, piperazine ring, fluorine, and ketonic group, which are responsible for its lipophilic nature. But these ionic groups like carboxylic acid or amines, these are responsible for its ionic character, and these are responsible for making it soluble into an water. So depending also once it gets solubilized into an water, once it enters into an lipid biomembrane, so these groups are responsible for binding with an DNA gyrase. So this ciprofloxacin, it inhibits an DNA gyrase, and these groups are responsible for binding with that DNA gyrase enzyme. And whether it will show acidic or basic properties, so all it depends on what type of environment it is having. When the favorable environment it is for an basic characters, so it will show an basic, or it will show an basic characters, and when environment it is favorable for an acidic condition, so it will show an acidic properties. So here in example we can see that now in the stomach pH it is acidic, so it is around 1 to 3, and this is a favorable condition for an basic to act, because in acidic conditions base will act, and in basic condition acids will act. So in stomach there it shows only an basic properties, because in stomach protons are been available, as proton pump is available in the stomach, and this proton pump it is responsible for an acidic conditions in the stomach, and protons which are available in the stomach, so it will show an basic action. When there is acidic environment, it will not show an acidic character, but as the pH changes, so it is more than 4 in case of an duodenum, so it is a part of a small intestine. Once, because in case of a small intestine there is basic pH, once drug entry into body through an GI tract, firstly it comes to an stomach, and from stomach it will enter into an intestine where there is pH difference. So basically it is said that stomach it is having an acidic pH, and after it enters into an intestine, so intestines are having a small intestine, it is having an basic pH. So after that you can see over here also that once pH it is more than 4, now it will show an acidic character also, because as the pH increases, or when there is favorable condition for an acid to act, so it will lose an proton from it, and it will show its acidic character. But when a drug it is in the stomach, it will show only its basic properties. So molecule containing multiple functional groups. So here we have seen alkyl aryl amine as an basic moiety over here, and carboxylic acid as an acidic moiety over here, so which are responsible for showing acidic properties as well as basic properties. And that is why the molecule it is said to be an amphoteric, because it is showing acidic as well as basic properties. So here in case of a duodenum where pH is more than 4, it is showing basic properties as well as an acidic property. So molecule is said to be an amphoteric, and which are responsible for ionization, or which are responsible for dissolution into the body fluids like waters, or in a serum, and other lipophilic groups which are responsible for its entry into a lipid bilayer. And this is how one should maintain during a drug design lipophilic and hydrophilic balance. So then only it will be ionizable, and then only it will be as it is an lipophilic, then only it will be entering into the lipid bilayer or different compartments of a body where receptors are ready for interaction, or where receptors are available for an interaction with a molecule. Now we'll see next concept, that is pKa. So it is said to be acid dissociation constant. So it is important to understand pKa, and it is related with the acid base properties of a drug. At whether molecule it is acidic or basic, or if it is amphoteric, so pH is responsible for its acidic as well as basic properties. So at acidic pH it will show an basic properties, and at basic pH it will show an acidic properties. So concept of pKa indicates the relative acid base strength of organic functional groups, and allows to calculate for a given pH exactly how much of the molecule is in ionized and unionized form. pK allows prediction of relative water solubility, absorption and excretion for a given compound. So all these are responsible for its ionic characters, because in case of an water solubility, molecule should be an ionic, and at the site of absorption, or to be an drug absorbed, so it should be in first and solution form, and then only it will be absorbed. So again here it's an ionic character. And lastly, that is excretion. So earlier also we have discussed that when drug it is lipophilic, so it will be not excreted. Only drugs which are hydrophilic, they will be only an excreted. So pK allows an prediction of all these characters. If the value of drug is equal to pH, then 50% of drug is in ionized form and 50% of drug it is in unionized form. So the pK, when it is equal to pH, at that time 50% of drug it has been ionized and 50% of drug it has been unionized. And this unionized drug, it enters into a lipid biomembrane, and 50% of drug which is ionized, so it enters into an water stream, or we can say a blood stream, which is having an hydrophilic characters. Now we'll see how pK works. And okay, now if we take an example of an functional group hydrochloric acid, so it is having a pK -8 and its conjugate base, that is Cl. So as pK increases, acidic properties it becomes weaker, and as pK is minimal, or as pK decreases, basic properties of the drug increases. So here you can see carboxylic acid having a pKa5 and its conjugate base. After that, protonated amines having a pK10 and its conjugate base. After that, water, OH, its conjugate base, and pK14. So alkyne. After that, amine with its conjugate base. So nearly the pK of this amine, it is 35, and pK of an alkane. So here you can see that as drug becomes more lipophilic, pK also increases, and pK is such a thing that 50% of drug and 50% of drug it is in the form of in an ionized form, and 50% it is in unionized form. After that, we will be looking towards a concept of an partition coefficient. So partition coefficient is one of an physicochemical parameter which influence drug transport and drug distribution, the way in which drug reaches the site of action and from the site of application. Partition coefficient is defined as the ratio of unionized drug distributed between organic phase and aqueous phase at equilibrium. So organic phase and aqueous phase, because body it is having a complex structure, to enter the molecule into lipid bilayer, so it should be in unionized form, and to get absorbed molecule, so it should be in a ionized form. So that is why partition coefficient it has been calculated using organic phase and aqueous phase. So here is a formula for a partition coefficient: concentration of drug in organic phase and concentration of drug in aqueous phase. As log P increases, so binding towards an enzyme's receptor also increases. aqueous solubility decreases. Binding to cytochrome phosphate 450 metabolizing enzyme, it increases. Absorbance through membrane also increases, and binding to blood or tissue protein also increases. So the meaning of this thing is that when the log P increases, so there will be increase in lipophilicity of a drug. As you can see over here, only an aqueous solubility decreases because ionization of a drug decreases as log P increases, because as log P increases, their molecule will become more lipophilic, and aqueous solubility it decreases. And all other properties like binding to enzyme receptors, binding to cytochrome phosphate metabolizing enzyme, or absorbance through membrane, and binding to blood tissue increases with a log P. But it should not happen that as drug it increases log P. So when it is been designed, so there should be a maintenance of an log P, or the maintenance of an lipophilic and hydrophilic balance, because aqueous solubility is also responsible for distribution and absorption of an drug. So what is importantly effect of an log P value on absorption and distribution? So we'll see through this chart. So here you can see the log P's value having -1, 0, 1, 2, 3, 4, 5 and 6. So when compounds are polar, so they are having log P values in this region. But when compound it is non-polar, so they comes in this category, that is from 4.5 to 6 almostly. And intermediate polarity, that is having lipophilic and hydrophilic balance equally. So these molecules will have an log P value around 2.0 to 3.5. When log P values are greater, so certainly a polar compound will show an good aqueous solubility, and this will show an poor aqueous solubility because it is lipophilic in nature. And when log P values are in this region, a molecule it is hydrophilic nature. So lipophilic over here after 4.5, and lipophilic over here, that is from -1 to almost 1. Certainly when compound it is hydrophilic, it will show an poor lipid solubility, and compound which is lipophilic will show an good lipid solubility. Poor absorption and distribution, while in between when it is having an lipophilic and hydrophilic balance, so it will show good absorption and distribution, but there will be slow excretion because molecule to be excreted, also earlier we discussed that molecule to be excreted, it should have a property of an hydrophilicity, or it should be easily solubilized with water. But as it is having a lipid property, or as it is having a lipid character, so it will be excreted. It will be not excreted, so it will move in the body in different compartments. But as lipophilicity, as hydrophilicity increases, so there will be excretion. Right? But due to less aqueous solubility, there will be slow excretion. So these are the compounds. So, or these are the log P values where compound suits easily. So you can see over here that it shows a good balance of aqueous and lipid solubility. So that it should not happen that compound should be only an liquid aqueous soluble and it should not be an lipid soluble, because aqueous solubility it is responsible only for a compound to get solubilized or to form in solution form. But this lipid solubility, it is responsible for compound to go into a lipid membrane. So this is how log P values affect. Now we'll see summary of today's lecture. So we have seen physicochemical properties of drug, introduction to it, and after that we have seen concepts of pharmacokinetics, absorption, distribution, metabolism, and excretion. After that, we have seen the concept of bioavailability, that is unchanged drug available at site of action. After that, we have seen the concept of solubility and dissolution, which are responsible for ADME. After that, we have seen acid-base properties of drug and pKa, acid-base property as per Lorry and Bronsted concept. After that, we have seen partition coefficient and effect of log P in drug designing. Thank you. Module number 4, lecture number 6.